Good afternoon, everyone. I hope you can see my screen and the audio is good enough. Please someone confirm.
Yes, sir.
Okay, thanks. So, good afternoon, and we will today learn about cloning vectors. Not vectors, but the cloning procedures. Until now, we have learned some tools, and in today's class, we are going to bring together all those that we have learned. And it's kind of review of whatever are there, and it is also going to be kind of primer for you about what kind of questions you can expect in the exam. Okay, so we'll go slowly and let's see what all we can do about it. So we will try to learn about how to use restriction endonucleases for cloning, blunt end ligation, linkers, adapters, and PCR primers. The first one is, of course, the simplest one, or the most rudimentary in a way, where we have the gene of interest, and the gene of interest should be cloned. Say, for example, it has something like that, and there are restriction sites, and we have a vector into which we are supposed to clone it. Say, for example, this is PAMH1 site, and the gene of interest also has PAMH1 site. And then PAMH1 restriction site is GGA, TCC, GGA, TCC. It will be ideal if you can also look into your, try to solve these things on a paper in your books. And say, let's see, if we select, one second. Upon restriction digestion, what will happen is that G, C, C, T, sorry. T and A would come about, and also this G. Okay, so the others will be, that is how the DNA would have been fragmented into upon restriction endonuclease, and it is going to leave. This is the 5' end, so this will be 3' overhangs. Sorry, 5' overhangs. Okay, so we cut the gene of interest on either side. We also cut the vector in here like this, and then we add DNA ligase. It's typically T4 DNA ligase, and then the gene of interest will be cloned here. And so here the purpose is cloning. What do you mean by cloning? In this place, this is, say, for example, the gene of interest. We want to make multiple copies of them using E. coli, for example. Then we can construct a recombinant DNA such as this, and we transform this into E. coli or whatever vector, whatever organism you may have had. E. coli is the most often used one. And once you grow the cells, say you take 10 to the power 6 cells, and from each of them you get 4, then you have 4 into 10 to the power 6 copies of your gene of interest along with the vector. This one. Typically, when you say gene cloning, it would mean that you are making multiple copies of them. And sometimes it's not just about cloning, you would want expression as well. In that case, you would have chosen a vector that is the expression vector, we call it, which would have a promoter for transcription to occur. And then you are supposed to clone it in the downstream of the enzyme, of the promoter. Please get your terminology very correct. what is meant by upstream, what is meant by downstream, and so on. Say if this is the reference point that we are talking about, then anything up is upstream, and this way it is the downstream one. Okay, and say for example here we have BAMH1 and EcoR1 restriction sites. And if we want expression to have happened, then we would want to clone it in such a way that the gene of interest should have been in this orientation. I already explained last time about what is this arrowhead, what does it indicate. This is the start codon, and the direction of reading of the open reading frame is indicated by this arrow. And this would have been the stop codon. So transcription should have happened from here to in this direction, so that the correct or the sense mRNA strand will be made, which upon translation by the ribosome will produce a protein. Okay. So now we should ideally have the gene of interest should be in such a way that we have BAMH1 and EcoR1 sites, or we should look at isosygomers that may be, or isocardamers that may be compatible. For simplicity, I will describe it this way. Say, for example, if this is the gene of interest and the start and stop codon are in this way, then you cut the gene of interest and also the vector, so that you will get overhangs, respective overhangs, and they are already compatible. And then you're going to put it in between in these sites here. Okay. So what you will then be able to do is, upon expression of the gene, you will get the required protein. So this vector would have been called as an expression vector. That is the topic of next chapter, so we will discuss it at that time. But otherwise, there are many a times we have the requirement for directionality of cloning, and therefore we should do that. The next one is filling up, and filling up as well as polishing. We discussed about it, and as I was telling you, there is, say I will cook up some restriction endonucleases, say for example GGA TCC and GGA TCC. If this is the gene of interest, then we have the restriction would have happened cut here and here. And if we have to separate them, they would have been separated in these ways. Let's see if I can make it happen. Okay, and this would have been here. So I hope you get the point anyway. It is a repetition, so you would already know it and see if you can. So it would have been like this. And as usual, there is 5' and this is another 5' end. And here is 3' end. This is 5' and this is 3' and this is 5'. So what we have now is something that can be used to, this will be the 3' OH, and DNA polymerase can be added, and then that DNA polymerase could fill up the gaps, GATC, GATC. And the enzyme that we use is called as clenofragment, which does not have the exonuclease activity. The DNA polymerase without the exonuclease activity, mostly the 5' to 3' exonuclease activity, is called as the clenofragment. So as in the previous case, if you have compatible ends like the compatible or restriction endonucleases that are helping you, then it is well and great. If it is not possible, then we may have to resort to other means. Even in the same case here we are choosing by default damage one site on the restriction site on either side and also on the vector. And then if it is there, it is well and good. If it is not there, what to do? Blunt end ligation is a good way of doing it. In some cases, there are restriction endonuclease sites are not matching, and still you have to clone it in directional ones. Those will lead to, we will need to enhance or try to use all the tools and see. Right now, I'll make a problem and I will send it to you across so that you can practice with that. So this is filling up using clinofragment. There are also other restriction endonucleases which would try to, I'm making it up, so don't take it literally. C A T A T G. Okay, just let's say I'm cooking it up. Okay, so if there was a restriction endonuclease that would have recognized this and cleaved it at certain sites, say for example here and here, if that is the point of cleavage, what it comes is CAT, AT, and G, and here we'd get G and TATAC. In this case, when it is the 3' overhang, remember, I hope you remember that I told you for DNA polymerase to fill up, you need a primer template junction and a free 3' OH and DNTPs. In this case, where there is 3' OH, there is no way of filling this gap using DNA polymerases. So what we should be able to do is chop off these DNA as well using exonucleases that have 3' to 5' exonucleases, single-stranded DNA exonucleases. And then we will be able to get these out. And now we have blunt ends, and then we can clone. There are, as I said before, when we want directional cloning, there are two approaches. One is you do a random cloning and see where you might get some of them, 50% chance of you getting in the orientation you want and you getting in reverse orientation. And in some cases, then in that case, you would then isolate the plasmids, construct the restriction maps, and then try to find out which one has the correct orientation and then work with it. This is also certainly possible, and one can do that always. Sometimes, wherever there is possibility, you would want to be in control, so you will favor the formation. If it is recombinant DNA, it should be the orientation that you are looking for. There are also other cases, some vectors when we will learn maybe. There are many different types of vectors, and it's innumerable in a way. There are promoters on either side of it, two different promoters. And if we have cloned it in this orientation, then we will try to use inducer for the promoter here. If it is cloned in the opposite one, we would have used inducer to induce expression from the downstream, this promoter. Okay? So it depends. There are many solutions to the same problem. So it's better to be aware of all these so that you will be able to employ anything that you might require. So the next one we learned is about linkers and adapters. Linkers, sometimes if there are none and you want to have linkers, you want to clone it, the gene of interest into a vector. And if these restriction sites are not matching, but you want it to clone in such scenarios, you can add linkers. Linkers are oligonucleotides which would have the restriction sites. I'm writing again for BAMH1, GGA-TCC. So this whole fragment you will add to the gene of interest. Okay, wherever it depends. It also many other things are there where you might cut this first from the DNA or the genome, and then see the compatible ends. Based on that, you would plan the overhangs of the linker, and then ligate it to the gene of interest. Once you do that, you are likely to get the gene of interest on either side with attached linkers like this. And then one can add restriction sites, restriction enzymes to cleave the DNA, cleave the restriction sites within the linkers, and then clone the ligate it to the vector. Okay, so that's with linkers. The adapters and linkers. Adapters, if you remember, I have given. If I want to have an adapter for EcoR1, maybe we should do it as an exercise. Let's see if we can make it. GATC, GGATCC, GGATCC. This is BAMH1, and I want to make an adapter for it. Why would you need one? Is especially if there is a restriction site like that within the gene of interest, and assume it is for BAMH1. Then in this step here, where we are going to add restriction endonuclease BAMH1 to cleave the adapters, it might also cleave the gene of interest in the middle if it were there. So in those cases, the best thing is to directly ligate just the adapters. And in that, I discussed about the chemistry of it a little bit, where I was telling you why there shouldn't be OH on the other side. One second. So if we have cleaved it, this part of the DNA will be lost, right? Overhang. And so is this. So on the sticky side, we want to dephosphorylate it so that it will not, this is 5' OH, sorry. This is 5' phosphate. Let me erase and write. So there will be some more of the DNA there, but we will just write it as this is 5' phosphate, and this will be 3' OH. And on the other side, it would have been 5' phosphate, but we would have used a dephosphorylation. We would have used alkaline phosphatase, for example, to dephosphorylate it, and then it will result in OH. So this is an important thing. So if I am representing it as a block, the adapter, I would put that this is the phosphate so that it can be ligated to the gene of interest, because the gene of interest would have 3' OH on one side. This is the OH of the adapter, and here we have the 5' phosphate. On this side, this is 3' OH of the adapter. And on the 5' side, there should have been a phosphate group, but we are going to remove it, and so there is an OH. So that is done essentially because we do not want self-ligation of the adapter, because we are going to add ligase, and if another adapter is something like this, it will be self-ligatory, self-complementary. If this was a phosphate group, right? Wait, I will just go back. So if you remember what I was telling you here, this will be the 5', and we made it as OH, dephosphorylated. This is OH. On here we have phosphate, and here we have 3' OH. Here also it is OH. So there is no possibility of a peptide, sorry, the phosphodiester bond forming on these sites. And that is an important thing when we are considering about adapters. I hope it is clear enough. Yes. And so this will be an important part of in the last class. I'm going to leave you for 10 minutes with this. It is 42 right now. I want you to design primers. I'm going to give you constraints, and quickly I will try to tell you the parts of primers. Primers should have parts of this homology, region of homology. It should have a restriction site, and it should have some additional bases for better interactions. So the constraints are, we will take the DNA whole of it. The region of homology, you take about 5 bases. Restriction site will be about 6 bases, and additional bases take 3 bases. Okay, this is an exercise. You please try it out and let me know when you are done, and post it in the WhatsApp group. I'll have a look at it, and then we will try to solve it again. So the constraint is, you have the vector is this way. There is the promoter. You have BAMH1 here, and you have EcoR1 site. BAMH1 is GGA TCC, and EcoR1 is GAA TTC. I think I have given you enough things. I want you to quickly draw the design the primers for this. You have 10 minutes to do that. I'm starting to answer it, just in case if nobody is trying. So what we'll do is, first thing, we have denatured the DNA, and I need to, first thing to remember is to whether to draw to take this sequence, primer design sequence, primers for this way or the other way, like this. What we should remember is polymerization always happens from 5' to 3'. So if I have chosen this, where if I have taken a complementary sequence here, say CTA, GG, and this would be the 5', and polymerization would actually happen only to the reverse side. So the blue ones are not converging, not good enough, right? So what is the best one is the red marks that I have put, where the sequence would have been, in that case, GATCC, which would have been 5' to 3' here. And the same way on the other side, CGAAC. This would be 5' to 3'. And then the polymerization, when it happens, it is going to converge. And that is when polymerase chain reaction can happen. Why is a chain? Because it increases exponentially if they are converging more and more templates. What the products of one cycle will become templates for the other next cycle. So if we see, the first part of it is done. I have taken regions of homology for 5 bases. I'll erase this 5' and 3'. And when you are writing the restriction site, it should always be on the 5' side. Okay, restriction site should be in this side, not here. The 3', okay, we would also try to draw up what are the important parts of the primer. And they have all primers, when you read from 5' to 3', they should have the region of homology. This is the region of homology. Even if you put 1 or 2 wrong bases here, then the primer is no longer useful. We will use that also. We will see some examples or alterations to that at a later time. Then it is followed by the restriction site. Right? And then we have these additional bases whichever may be there. So when I'm writing the primer, I have to take the restriction site in such a way that it comes to the other side of it. I will write it in blue. So, and when you're writing the restriction site should have been read in 5' to 3'. And also I gave you a constraint that it should be BAM H1 and EcoR1. One of the downstream, the order of downstream of the promoter should be BAM H1 followed by EcoR1. So that means you want your gene of interest to be that way. So we have to write restriction site GGATTC. Okay, and on the other side, we should not write it GAA like this, because it seems like that here, right? So ensure that you're reading it properly, and you would be writing it GAATTC. Always the restriction site should be read from 5' to 3' and still give the sorry, wait, I made a mistake here, I guess. So it'll be easier if you are writing it 5' to 3'. Otherwise, it's always confusing. Right. So here, what we should do now is, if I'm reading it in this direction, 5' to 3', then I have to put EcoR1 site in that particular orientation. Then GAATTC. Okay. Restriction site should be that way. And then we will have the additional sites, could be any of them. You can add something CCC or so. And now when polymerization or PCR is completed, what we will find is, okay, let me verify if the if the primers are sticking to the skeleton that I have drawn here. Or this is usually called as the forward primer, but otherwise different labs can have it differently. And this one will be the reverse primer. So on the 3' side, there should be region of homology. Then you have the restriction site, properly oriented 5' to 3', and then additional bases. Let me look at this here. The reverse primer, region of homology is on the 3' side, and recognition site is GAATTC. It is being read correctly, and then the additional bases. Okay, and once we perform PCR, yes, please.
Sir, I have a doubt regarding that additional basis. Like,
Yeah.
is it like mandatorily CCC or like?
No, you can use any of them. Usually, though, you can choose CCC because at the ends, okay, they will have triple bonds and they will have G, right?
Yes, sir.
So they're likely more stable because of at the ends. But otherwise, it doesn't matter. People add. I have added different random sequences also. I have added different restriction site also sometimes, just like that. So it's not. It's all right. There is a lot of liberty in that.
Okay, sir. So, okay, just for the purpose of the question, we'll just give it a CCC for.
Yes,
Perfect,
but
sir.
otherwise it can
We
be.
don't need to avoid confusion.
Yes, yes.
Okay, sir.
Yes, good. So once that is done, then we will have the, upon PCR, what we will get is, we will get a product. I'm not writing the sequence anymore. We will get this is the product that we are looking at. And then we have the restriction site. That is, on one side we have BAMH1, on the other side we have the EcoR1. And then the additional sequences. Once we finish this, we will digest it with BAMH1 and EcoR1. So we will get these. The vector can be cut into, and also the fragment or the PCR product will be cut on either side. And then we will have this. And that is compatible BAMH1 here and EcoR1 there. So this will ligate here. I'll put it in different color in this orientation. Of course, here I did not check for if there is an ORF or not. I have just given you that I want it to be cloned in this direction. So I'm just putting it this way. EcoR1 and BAMH1. And this should be ligate. This will be ligated in that way because the complementary, the ends, compatible ends would base pair. Is there any question here with the PCR primers? Primers. We have other enzymes that we did not discuss, is probably alkaline phosphatase. It is used for dephosphorylating the 5' ends. We have also used terminal transferase, that is for homopolymer tailing. What else? Are there any other enzymes that we missed?
Sir, did we include DNA ligase or is it included already?
Yeah, okay. DNA ligase, T4 DNA ligase is one thing, and E. coli ligase is also there. Most often it is T4 DNA ligase. We also have modification, say M-BAMH1. That is the modifying enzyme of BAMH1. It would have if there was an internal site here of BAMH1, and if you specifically can methylate it, then you can use M-BAMH1 to methylate this BAMH1 site. Are there any other enzymes that we have missed? I think that is about it. Okay, in the next class onwards, we are going to, let me have a look at it again so that I'll ensure everything is done. Yes, so the reason for why I have taken this up again is because we are going to start the next chapter that's on vectors. And I will just briefly introduce and leave you with that, okay? I'll not start the whole lesson as such. What is a vector? In different, if you are looking at biology, you will say for diseases, you will see that some pathogen is there, and that pathogen grows in some organism, and that might bite a human and cause disease. This organism is referred to as in that scenario as vector. Vector is something that carries, right? In this case, all the examples that we were discussing here, this is our gene of interest. And we are putting the gene of interest into a DNA molecule, and that is carrying our gene of interest. So we call that as vector. So there are many different types of vectors. Vectors could have been derived from plasmids. They could have been derived by phages or even chromosomes, bacterial as well as eukaryotic. Okay, and there are so many varieties of them. We cannot cover all of them, but we will cover some of the very important ones. How do I describe what is important and not? For me, what is important is that will allow you to use your analytical thinking. If that is there, I would like the vector. As a matter of fact, every vector, and because this is an applied science in a way, so every vector is made with a certain purpose, and the purpose has a logic, and our job is to know the logic and be able to manipulate the logic or employ that logic in other scenarios. So we will start off with general features. You are already studied with them, and some of you haven't studied, so I will take it from basics. A vector should have multiple cloning site or a cloning site. That is, there's a series of restriction sites here. They should have a selectable marker. We will define them again, and we will try later in the next class. They should have a selectable marker. And then they should have maybe a promoter for expression. The most important thing is that they should have origin of replication. If there is no origin of replication, it is just a DNA. It is not a replicon which cannot be replicated. You can take any of these plasmids, phages, chromosomes, any source of DNA from naturally. If they are propagating within the organism, that means they are undergoing replication. For replication to happen, other than the other substrates and enzymes required, there is an element, genetic element that should be there within the molecule, is the origin of replication. Origin of replication is the site where replication initiates. enzymes are designed to start replication in those particular locations. Replication does not happen everywhere, right? So there are replication in throughout biology. It is essential and it is highly orchestrated. That means it should happen in a defined, regulated ways. So the origin of replication will allow the replication of that molecule. If we cut this out, if this origin of replication is not there, then nothing is going to happen to it. It will remain same single copy. It may exist as long as it is. It is not going to form more copies, and there is no cloning then. Okay? And if one cell is going to become two cells, right? And the one of the cells has the plasmid, the mother cell. And if there is no replication, only one cell will get it. And if there are further rounds of replication, say one cell has formed a colony of some 10 to the power 10 cells, and that one cell had a plasmid, but without origin of replication. At the end of the colony formation, there will only be at the most one cell with that with vector, with that plasmid, and all the other cells, okay, all the other cells will not have had this plasmid if origin of replication is not there. If origin of replication is there, then it's likely that most of the cells may inherit it. Okay? And selectable marker, I will define what is meant by selection. Okay? And selection is something where, maybe I'll take it in the next slide. What is selection? Say, if there are some cells, or say I could have put it in shapes maybe. One second. Let me try a different example maybe. I'll try these squares, some big ones and some small ones. Assume there are many of them, small ones. Now I need to have a way of selection. And the selection is that I want only the big ones here. Okay, then how will you have to do one procedure, one activity, because of which you will get only the big ones, big squares, and you would have gotten rid of all the small ones. What would that be? Say if there were something like grains, you would have used a sieve-like thing, right, with holes big enough so that big enough for the small ones to pass through, and big enough for the small squares to pass through, and small enough for the large ones to be on the surface, right? That is the activity. You're using a sieve to just get off the small ones. That is selection. The same thing can be if you are trying to do it in biology, in sense of biology. Say, for example, you had several bacteria. You have many bacterial cells, and you want to select the one that is resistant to ampicillin. Then you would have plated them all on agar plates containing ampicillin. Okay, the nutrient agar should have had ampicillin. Those that are sensitive to, those that have resistance to ampicillin would survive, and the rest of the cells would die. And those that are resistant would have made colonies. You will get 3 colonies, assuming those are the number of things. So you are selecting with these organisms with the trait. So basically, or simplistically, if you're talking about selection, it is an activity or a procedure, you can say, to obtain objects or cells here with required traits. Right? So that is selection. If I had so many cells and assume that the plasmid is the vector that I'm talking about is in some cells, usually when you do a transformation experiment, say for example, you take 10 to the power 6 cells, and maybe about 1,000 of them would get transformed, or usually say 10 of them got transformed. How can we select them? So you can't take one cell at a time and see whether whether the plasmid is there or not. That's a very difficult procedure. Instead, if we can have a selection procedure where the 10 will grow and the rest cells would die, and that is for this procedure to be for this selection method to be employed, we should have had the vector should have a selectable marker. Most of them are antibiotic resistance genes. Okay, but there are also other biochemical ones or synthetic protein genes and other things are also possible. We will see about them at a later time as well in the next classes. So I hope I gave you enough to think about and be prepared. I have reviewed whatever we have discussed so far in the course, and I hope you have some idea about the every topic that we have covered so far. Okay, if you have any questions, please let me know now. Stop the recording here. How is the assignment coming?